Hello, welcome to e-learning. I am Dr. Gauri Abhyankar, working as Assistant Professor in Abasaheb Garware College, Pune. This session is the second part of chloroplast and mitochondrial study. Here we will deal with biogenesis, that is formation of these organelles, their multiplication and evolution. The chloroplasts are considered to be originated from a free living cyanobacteria like prokaryote through engulfment over 1 billion years ago. Each chloroplast arises from a pre-existing chloroplast by binary fission. Chloroplast is surrounded by an outer envelope membrane and an inner envelope membrane like a prokaryotic cell. In higher plants, chloroplast can differentiate into various non-green plastids. Plant mitochondria are complex organelles. They carry out various metabolic processes related with energy generation as well as synthesis and degradation of several compounds. Mitochondria are semi-autonomous and dynamic organelles. The learning objectives for this session are to study formation and development of chloroplasts and mitochondria. Secondly, to see the multiplication process of chloroplasts. Then, we will see how chloroplasts have evolved. Finally, to study mitochondrial division, fusion and fission in detail. Chloroplast biogenesis is a multi-stage process which leads to fully differentiated and functionally mature plastids. Chloroplasts develop from proplastids present in leaf primordia and possess small vesicles without any photosynthetic complexes. On exposure to light, proplastids develop the thylakoid network and achieve photosynthetic capacity. You can see a schematic overview of light dependent deetiolation process and biogenesis of thylakoid membranes, focusing on pigment system 2, 1 and light harvesting complex. In dark conditions, proplastids mature into etioplasts. Inner membrane of etioplast forms a prolamellar body which is semi-crystalline network of interconnected tubules. The prolamellar body produces extensions and forms prothylakoids. Because they contain lower amounts of monogalactosyl diacylglycerol, that is MGDG, they permit them to form a planar bilayer structure that is seen in figure A. The prolamellar body is photosynthetically inactive. By now, ATP synthase is fully assembled. Cytochrome B6F complex is present as a dimer. The most abundant protein in prolamellar bodies is NADPH protochlorophyllide oxidoreductase. Figure B shows etiochloroplastic stage. The deetiolation process is initiated by exposure to light. Upon exposure to light, protochlorophyllide is reduced to chlorophyllide by POR and finally esterified into chlorophyll. At the same time, as you can see in figure C, the prolamellar body loses its semi-crystalline structure and the extruded lamellae align in parallel throughout the stroma. The semi-crystalline prolamellar body then becomes planar thylakoids. The grana stacks characteristic of mature thylakoids form upon incorporation of mega and super complexes. Chloroplasts are found in different plant organs depending upon the species and stage of development. Due to tissue specialization, biogenesis and development of chloroplasts differs between organs as well as between species. Tight assembly of nuclear encoded proteins as well as chloroplast genes along with chlorophylls and carotenoids is very crucial for photosynthesis. This requires coordination between the two organelles at many different levels like transcription, translation, import, protein turnover and metabolite flux. These we can call as plastidic and nuclear factors required for biogenesis and development of chloroplasts. First factor is nuclear gene transcription. When the seedlings receive light, most of the genes encoding chloroplast targeted proteins are upregulated. The light mediated transcription requires involvement of phytochrome interacting factors, and their loss results in delayed development of the chloroplasts. Second factor is chloroplast protein import and processing. Most of chloroplast targeted proteins enter the plastids via the translocon at outer or inner envelope of chloroplast. The proteins are transported to translocons by their chloroplast targeting peptide. If it is knocked out, then chloroplast biogenesis is dramatically impaired. Next factor is chloroplast gene transcription and translation. Chloroplast biogenesis and development are dependent on chloroplast gene transcription, RNA maturation, protein translation and modification brought about by both nuclear encoded polymerase and plastid encoded polymerase machinery. Loss of the plastidic transcription factors delay chloroplast biogenesis. Protein folding and degradation is another factor here. Chaperones mediate chloroplast protein folding. Mutation in these chaperones leads to variegated cotyledons. Next factor is thylakoid formation and pigment biosynthesis. The thylakoids are formed mainly by mono and digalactosyl diacyl glycerol. Carotenoid and chlorophyll biosynthesis is tightly regulated during chloroplast development because they are essential for the assembly of photosynthetic complexes and photoprotection. Last factor is retrograde signaling. Most of the proteins required for chloroplast function are encoded in the nucleus. Various factors are involved in the control of retrograde signaling. Along with these, there are also certain non-plastidic factors like environmental, cellular and temporal factors that are essential for chloroplast biogenesis and development. Environmental factors involve light as a major factor. Cellular factors include the communication between the organelles. Embryo development is one of the temporal factors that influences chloroplast biogenesis in germinating seedlings. Chloroplasts divide by binary fission, as could be seen in moss Physcomitrella, but in certain cases they show multiple fission. Localization of the FTSZ protein at the chloroplast division site in Arabidopsis is also observed in next figure. The plastid division apparatus is basically evolved from the cell division apparatus of cyanobacteria. This was postulated when homologs of the bacterial cell division protein FTSZ was observed during chloroplast division. General overview of plastid division can be observed in the right hand figure. Here is the process of chloroplast division. FTSZ is a tubulin like cytoskeletal GTPase and is able to self assemble spontaneously into Z ring at the division site. Once Z ring is formed, assembly of the cell division complex starts. The assembly helps for the constriction. Similarly, Z ring is formed at the equator in chloroplast division. This may initiate the assembly of the plastid division complex. Plants and green algae have two nuclear encoded forms of FTSZ, namely FTSZ1 and FTSZ2. Both are components of the plastidic Z ring and function in plastid division. FTSZ2 is more similar to its bacterial ortholog and is able to assemble in absence of FTSZ1. Combined activities of both these factors control the positioning, assembly and disassembly of the Z ring. Symmetric division in both plastids and bacteria is achieved by placing the Z ring in the center. Two key candidates FTSZ regulators in Arabidopsis are ARC3 and ARC6. ARC6 is a regulator of Z-ring assembly and dynamics. It is homologous to the cyanobacterial cell division protein FTN2. Chloroplasts share common ancestors with existing cyanobacteria. Chloroplasts in green algae and mosses usually do not develop from or into non-green plastids. Mitochondrial biogenesis is the process of formation of new mitochondria in the cell. As they arise only from pre-existing ones, mitochondrial biogenesis involves different mechanisms related to synthesis and assembly of new mitochondrial components. The figure shows integrative view of different regulatory pathways involved in mitochondrial biogenesis. In first regulatory pathway, mitochondrial biogenesis is regulated by numerous external factors. Secondly, it is also internally regulated by different stages of development, organ or tissue type, hormones and cell energy and metabolic demands. Third is the control of expression of a majority of nuclear genes for mitochondrial proteins by site 2 elements. They are either responsible for basal gene expression or modify the magnitude of the response under different growth conditions. Next is dual targeting of proteins to different organelles, which may act to coordinate the activities of these organelles. Next pathway shows how physical and functional interactions between the inner membrane, import machinery and complexes 1, 2 and 4 help to adjust gene expression and protein assembly, and finally, last pathway depicts how mitochondria generate signals to modify nuclear gene expression according to organelle demands. Now, let us move to mitochondria. The figure shows you the mitochondrial division. In all types of cells, mitochondrial division maintains the proper cellular distribution of mitochondria. In yeast and in higher eukaryotes, mitochondrial division occurs through DNM1 and its homolog DRP1 respectively. They belong to a family of dynamine related proteins. Dynamine related proteins are large self-assembling GTPases possessing highly conserved GTPase domain. As it is observed in the figure, DNM1 in yeast is localized to the mitochondrial surface, whereas in higher eukaryotes it is diffusely dispersed throughout the cytosol and a small fraction of the protein is found assembled on mitochondria. The current working model of mitochondrial division involves DNM1 or DRP1. The process can be broadly classified into 3 stages. First is targeting, second is assembly driven constriction, and last is hydrolysis mediated constriction or scission. The steps are as follows. Initially, DNM1 or DRP1 is targeted to surface of mitochondria. Then it undergoes GTP driven assembly into helical structures. Next, this self assembly drives constriction of membrane or mitochondrial tubule. GTP driven assembly also stimulates GTP hydrolysis. GTP hydrolysis induces conformational changes within DNM1 or DRP1. The helix required for further constriction. Membrane division is completed now. We know that mitochondria are dynamic organelles that are constantly undergoing both division and fusion. Mitochondrial fusion is a highly conserved process from yeast to human cells. Mitochondrial fission contributes to proper distribution of mitochondria in response to the local demand for ATP. It also helps for elimination of damaged mitochondrial fragments through mitophagy. Mitochondrial fusion and fission in yeast are controlled by 3 high molecular weight GTPases, namely FZO1, MGM1 and DNM1. DNM1 is mainly found in the cytosol. During mitochondrial fission, it is translocated from the cytosol to fission sites on the mitochondria. DNM1 or DRP1 forms self-assembled higher order structures that wrap around the mitochondrial tubule. These spiral structures squeeze and eventually cut off the mitochondrial membrane. The process is dependent on GTP hydrolysis. To summarize the session, we learnt in detail about biogenesis of thylakoid membranes and various factors influencing the process. Then we have discussed about the process of chloroplast division. We have studied mitochondrial biogenesis and the regulatory pathways for the same. We have detailed out the model for mitochondrial division. At the end, we talked about mitochondrial fission.